Hello and good morning, everyone. Thank you for joining us for today's presentation in our 2022 Covey Lecture Series. My name is Doug Hunt, and I'll be your moderator today. Our lecture will run about 45 minutes, and then we'll have a question and answer period following that. Please feel free to type your questions into the chat feature at any time, and they will be shared at the end. I'm very pleased to welcome Covey Director and researcher Debbie Ingles as our speaker today, and she will be talking about enhancing the Sauvignon Blanc character in Vidal table wines. Debbie attained her PhD from McMaster University in biochemistry in 1995. After a 3-year stint in the biotech industry, where she did her postdoctoral fellowship and then worked as a senior scientist in R&D, she joined Brock in 1999 as an assistant professor in the Department of Biological Sciences. She started teaching in the Enology and Viticulture program, as well as in biochemistry. She took on the directorship of the Cool Climate Enology and Viticulture Institute in 2008. She now holds the title of full professor and continues to be director of Covey. Debbie's research program first focused on issues of ice wine fermentation, but has expanded over the years to other wine styles, such as appassimento and sparkling wines, tannin management in red wines, sour rot management in grapes and wine, grapevine cold hardiness, and the biochemical changes occurring in the vines during winter dormancy, and, more recently, diagnostics for grapevine diseases. Debbie has been involved in the grape and wine industry her entire life, as her family were grape growers in the Niagara Peninsula, and her brother owned a winery. She is a second generation grape grower and was crowned Grape King for Ontario in 2010 based on vineyard excellence. She and her husband Rob still run their own vineyard in Niagara-on-the-Lake. So with that, Debbie, I'll ask you to please go ahead.
Thanks, Doug. Thanks for the introduction. Yes, just listening to all that background information, I've been around for a little bit here. So today I'm going to actually talk to you about a new project that we started a couple of years ago on enhancing the Sauvignon Blanc character of Vidal table wines. So to start off, I just wanted to talk a little bit about the history of our industry. And the industry in Ontario now has a 4.4 billion economic impact for the province. We have over 18,000 acres. We're dominated in production by Vitis vinifera varieties. And we're really striving for, that VQA industry to support Ontario-grown grapes. The crop in 2021 was worth over $100 million and brought in over 82,000 ton. And this is from the most recent GGO annual report that was just presented in 2022. So of that tonnage, 62% is made up of Vitis vinifera, and about 38% are made up of hybrids. But when we look at the most dominant hybrid that we grow in the region, being Vidal, it makes up almost 25% of the total grape production in Ontario. So it's an important variety for us. And it's important for a number of reasons. It is a more winter hardy variety than the typical Vitis vinifera varieties that we really base our industry on. It's used extensively in ice wine production, where Canada is the world leader by volume in ice wine, and I would argue by quality. But Vidal can also be used on its own or in blends in table wine production. Now, the industry is still challenged with cold weather events that can severely impact annual grape production. And if you need any evidence of that, if you go to the VineAlert site for 2022, you'll see that January has taken its toll in some regions across Ontario in terms of bud survival for Vitis vinifera varieties. Now, Vidal is a hardier white variety in comparison to Vitis vinifera varieties, where it can better survive the erratic cold weather temperatures and swings of the winter months in the Niagara Peninsula, and continues to be a fruitful producer year in and year out. So the variety offers assurances to grape growers of a crop and income after less than ideal winters where Vitis vinifera varieties can suffer. And so because of this, there's still a lot of production of Vidal, and growers rely on Vidal for sustainability reasons for their operations. Now, Vidal is the leading grape variety used in ice wine production, but ice wine markets are in decline the last few years, and especially with COVID, they've really taken a hit. So the industry as a whole is looking to diversify the use of Vidal in table wine because we have so much of it. We grow so much of it in the region. The grapevine itself is relatively resistant to damaging cold temperatures. The fruit ripens consistently in our cool climates, and therefore, as I mentioned earlier, it offers stability in grape production to the grape growers, as well as to the wineries. Now, our project is looking at enhancing the aroma profile of Vidal table wine. And my colleagues and I that are working on this research, Dr. Jim Woolworth from Biological Sciences and Covey here at Brock, and Dr. Amy Bowen, a research director in consumer sciences over at Vineland Research and Innovation Center, we all have anecdotal evidence from tastings of commercially produced Vidal in Ontario, in Quebec, and Nova Scotia, that for a handful of these wines, we noted that sometimes these wines really show characteristics of Sauvignon Blanc. And I further had some discussions in 2019 about this with a local grower, one of our partners on this grant, Albert Zager. And after tasting some of his homemade Vidal, we were also picking up this Sauvignon Blanc character. And we really wondered if these aromas and flavors in these Vidal wines could be due to the aroma and flavor compounds in Sauvignon Blanc that give it its characteristic nature. And these compounds are known as volatile thiols. So what are these volatile thiols? Well, there's 4 main ones that are found in wine, and their chemical names are here in this chart: 4-methyl 4-mercaptopentane 2-one, or 4-MMP, that I'll short form it to, 4-methyl 4-methylpentane 2-ol, or 4-MMPOH, 3-mercaptohexanol 3-MH, or 3-mercaptohexyl acetate, the acetate ester of this alcohol, known as 3-MHA. And when you look at the sensory descriptors of these compounds, you start to see the linkage of what we were finding in some of these Vidal wines. Many of these volatile thiols have aromas of plants such as box tree and broom. These are the 2 plants I've put some pictures of up top. Black currant, cat urine if it's at a high enough concentration. Often they have grapefruit or passion fruit aromas and flavors, and even gooseberry as part of the profile. So we were picking up, these aromas, these grapefruit and gooseberry aromas, in these Vidal wines, and wondering if they could possibly be due to these volatile thiol compounds. In 2017, there was a publication that came out on Vidal ice wines made in China. And one of these dominant volatile thiols, 4-MMP, were showing up in these wines. Now, this is the chemical structure of these compounds. I'm not going to focus too much on them, but why they're unique and in a family is due to this thiol group, this SH group, sulfur hydrogen or a sulfhydryl group that is common in all of these volatile thiols. Now, the 4-MMP-OH, the first one here, is generally below detection thresholds in wine, but the 4-MMP, the 3-MH, and the 3-MHA are often found and detectable in Sauvignon Blanc wines. So we really wanted to look at whether we could enhance the Sauvignon Blanc character of Vidal. Could market demand for Vidal table wine be improved through enological and viticultural treatments to make a wine more competitive and desirable for the consumer? We were really hoping that if we could help improve the aroma profile of these wines, that these wines as a table wine could compete with foreign imports and offer a lower price option, probably around that $12 a bottle range. What we know is that consumers are increasingly seeking local products, but they still need to be of high quality and affordability for consumers to switch over. So what we were looking at with Vidal to try to enhance that volatile thiol level in the wines was really drawn from research that we know and is published on Sauvignon Blanc. So in Sauvignon Blanc, we know that manipulating crop load can change the volatile thiol content in the wines. So we wanted to look at if we reduce the crop load to 6 ton per acre versus a typical 10 ton per acre, would we see a difference in these volatile thiols if they were present in the wine? We also wanted to look at the maturity level of the fruit, going with 18 degrees Brix or less mature fruit versus 22 degrees Brix of sugar or a more mature cluster of grapes. Would that alter the volatile thiols? We also know yeast strain can have an impact on producing these volatile thiols. There are precursors in the juice that will get taken up into the yeast cell, and then the yeast modify these precursors and produce these volatile thiols and then release them into the wine. So I contacted Laumon, and they gave me 3 suggested yeasts that are they recommend for further enhancing the volatile thiol production, and that was QA23, IOCB thiols, and the Sauvy yeast. And then they also recommended to me that we use a nutrient additive known as Sauvignon Blanc Stimulate, which supposedly provides these precursors to the yeast that would then allow them to take these compounds up and produce more of these volatile thiols. So we laid out the experimental design, myself and my colleagues, Jim Woolworth and Amy Bowen, to look at a scientific evaluation of varying these factors that can improve the expression of Vidal character in table wine and improve its market preference with consumers. So the multiple factors that we wanted to vary were the crop load of the Vidal, the maturity level of the grape via that Brix measurement at harvest, and the yeast strains used during fermentation. But we also, through the work of Dr. Amy Bowen at Vineland, wanted to do consumer liking trials with a consumer panel to determine what treatments of these Vidal research wines had the highest consumer appeal, and what would consumers be willing to pay for these wines. So again, this was very much a collaborative project with Dr. Woolworth focusing in on the viticulture treatments. I oversaw the enology treatments, and Dr. Bowen was focusing in on the consumer liking and willingness to pay research. So in all, we had 12 different treatments that we wanted to investigate, and each treatment was fermented in triplicate, so that gave us 36 fermentations. And all of this work was carried out by a postdoctoral fellow working in my lab, Dr. Jen Kelly. So we divided the work into the low crop treatments at 6 ton per acre and the high crop treatments at 10 ton per acre. Then at each of the low and high crop values, we had two different harvest dates, when the grapes reached 18 Brix and when the grapes reached 22 Brix. And then for the fruit at 18 and 22 Brix, we fermented them each with 3 different yeasts, the QA23 yeast, the IOC thiols, and the Sauvie yeast. Now, I just want to share with you some of the juice data prior to fermentation. And you'll see from this chart that we weren't too far off with the low Brix treatment. We were targeting 18 Brix, and we were between 17.5 and 17.8 Brix in 2019. So we were a little shy of our target, but not too far off. However, we continued to let the grapes hang and hang and hang, and 2019 was just not a very warm year. And the grapes just really only increased by just over a Brix in sugar level. So our high Brix treatment, which we were targeting 22, really only went up to about 19 Brix. So we weren't really seeing the maturity difference in our treatments that we had hoped for. So we'll just keep that in mind. Also, when we ran the nitrogen values on the juice, they were a little bit low in nitrogen. So we decided to supplement all the ferments with diammonium phosphate, adding an additional 50 milligrams of nitrogen per liter to ensure that the yeast fermented to dryness and that there was not going to be any problems with a stuck fermentation or hydrogen sulfide production. But I'll come back to that part a little bit later on because we think this is an area we can further manipulate to further enhance the volatile thiols. Also, just to point out that the grapes were fairly acidic and that carried through in the wines. So in terms of the fermentations, when we looked at first the low crop treatment at both the lower Brix target, 18 Brix, and the higher Brix target, meant to be 22, but remember it was only about 19. What we found was that in both cases, the QA23 yeast lagged a little bit behind in its sugar consumption rate in comparison to the Sauvie and the IOC thiols. However, really by day 8, all the yeast had fermented to dryness, and we really didn't see a huge impact on the Brix difference for these treatments. Similarly, in the high crop treatment, so this would have been the 10 ton per acre fruit, again, very similar profile for how the yeast performed, QA23 lagging behind again, but by 8 days into the ferment, all the yeast had fermented to dryness. So a little bit of difference, but I don't think enough based on fermentation performance that anyone would really be concerned with QA23. But what we were really excited about when we went to look at the volatile thiols is that in the Vidal wines, and these are the wines with all the various treatments in the blue and the green, we were picking up 4 MMP and 3 MHA. And the values were quite similar to the values that we were finding for a New Zealand Sauvignon Blanc that we ran just as a test control. So in the New Zealand Sauvignon Blanc, you can see that the values here for 4 MMP and 3 MHA are quite similar to the values that we were seeing in all of the experimental treatment wines. However, one big difference, we were not picking up the 3 MH in the Vidal wines, but we were picking that up in the New Zealand Sauvignon Blanc. Now, the 4 MMP, you'll remember, provides that broom and box tree, blackcurrant aromas, maybe a little bit of cat pea urine there as well. And the 3 MHA, those are the fruitier compounds like the grapefruit, the passion fruit, and the gooseberry. Additionally, what we were finding, specifically with the 4-MMP concentrations, we routinely were finding that the Sovi yeast was producing a higher value of 4-MMP compared to the B-thiols as well as the QA23. And you can see that by the different letters that we have beside the concentrations of these compounds. When they're different letters, that means that the values are statistically different from each other. Whereas you can see in the 3-MHA values, they all came in about the same level for all of the different yeasts. So what we were excited about here is that when we were measuring volatile thiols in the Vidal wines, that we were seeing some yeast differences in the 4-MMP, but what we weren't picking up were any differences in the values from crop level or from that grape maturity. But again, remember that the grapes were only differing by about a brick in 2019. So we wanted to continue on with this study in 2020. It wasn't part of the original design to do fermentations in 2020, but when COVID hit, we were somewhat restricted in our time allowed in the lab, and our grape grower partner, Albert Zager, agreed to give us some additional grapes so that we could carry out ferments again in 2020. And 2020 was a warmer year compared to 2019. And you can see here that we were able to get grapes of 2 different Brix values, about 17.5, which was targeting our 18 Brix value, and 20.6, close to 21, for our 22 target. Now, because we weren't in a position to do all the treatments over again, we just focused on the high crop treatments. So we didn't go through and thin the fruit to get the low crop. So this would be more representative of what's typically grown in the region. Similarly, the nitrogen values were a little bit low, so we supplemented with DAP, and we decided this year as well to throw in a control from EC 1118 yeast. So this is just a workhorse yeast used in the industry. It's not known for producing high levels of volatile thiols or anything like that. So we wanted to include it just as a baseline measurement for us. So again, when we carried out the fermentations, once again, we found that the QA23 yeast lagged a little bit behind the other 2 yeasts. In the 18 Brix treatment, not quite as noticeable in the 22 Brix treatment. The IOCB thiols in 2020 fermented at the fastest rate and really completed the fermentations by about 7 days. And the others, again, needed about an extra day to complete. We did find the EC 1118 fermented the slowest out of all of those yeasts that we tried, and it took about 10 days to complete the ferment. So when we looked at the volatile thiols in these wines, here we were seeing a difference in the 4-MMP between the 18 Brix treatment, which came in at about 17.5 Brix, and the high Brix treatment, which again for us came in at about 20.6 Brix. So we were seeing that the riper fruit enhanced the 4-MMP concentration a little bit further. And once again, we saw with this Sovi yeast that they produced the highest level of 4-MMP, and that was true whether it was at the low Brix treatment or the high Brix treatment. And our values for 4-MMP were also coming in higher than what we were finding with a control New Zealand Sauvignon Blanc that we measured. Once again, in that New Zealand Sauvignon Blanc, We were able to measure 3MH, which provides some of that fruity grapefruit characteristic to the wine. We were not able to detect that in any of the Vidal wines, but we were able to detect the acetate ester. And the acetate ester values came in quite comparable, even a little bit higher than what we were finding in the New Zealand wine. So the summary from this is that the later maturity high Brix fruit led to higher 4-MMP values in the wine. The Sauvy yeast, once again, had the highest concentration of 4-MMP, and it was higher than the New Zealand wine that we tested. And the Sauvy and the IOC bethiols were similar in 3-MHA, and the QA23 was slightly lower in one of our treatments. And no 3-MHA was again detected in any of the Vidal wines. So this led us on to then do the sensory and consumer testing on these wines that was carried out by Dr. Amy Bowen and her team at Vineland Research and Innovation Center. So when her team evaluated these 12 different treatments, they used a rapid profiling technique to describe the aromas and flavors that they were picking up in these wines. And she had a panel of 13 trained panelists that evaluated these wines. And out of those 12 wines, it grouped the wines into 5 different groups. And these are the representative descriptors of those 5 groups. And you can see we've got descriptors as chemical and citrus and acidic, to rubber and tropical, to acidic, flory, earthy, tropical, and yeasty and flory. And I'll just point out to you this last yeast, which is the Sauvy yeast, which was described as gooseberry, citrus, and tropical. So these groups of wines were then further put forward to a consumer panel to test with the consumer panel what their preference was, what their liking scores were for these different wines. And when we did the consumer liking scores, or I should say when Dr. Bowen did this work with Dr. Kelly and Dr. Bowen's team at Vineland, they also included a white wine blend, which was a Vitis vinifera blend, in the price point that we were trying to target these wines, you know, around that $12 a bottle. We also included 3 commercial Vidal wines, along with a wine that was representative of each of those 5 groups that the sensory panelists had defined. And what we found was that the white blend wine had the highest global liking score. But what we also found was that the Vidal wine made with the Sauvy yeast was 3rd in ranking, but statistically not different from the white blend. So whenever you see the same letter beside a number, it means that there's not a statistical significance in difference in consumer liking score between those wines. We found that one of the wines made with the Bethiols IOC yeast and the Vidal 3 wine grouped into a different consumer liking score. And so these were lower on the scoring, significantly lower on the consumer liking scale compared to these other ones. So this was pretty exciting news for us as well, that our research wines, 4 out of the 5, all grouped together with that white wine blend that the consumers liked. Now, Dr. Bowen took this a little further to better understand what the consumers were detecting and liking in these wines. And this broke the group out into 3 different groups: Group 1A, Group 1B, and Group 2. And you can see Group 1A, they had higher liking scores for wines that were defined by fruity flavor. And they had a higher liking score for the Sauvignon strain compared to 2 of the commercial Vidal wines, Vidal 1 and 3. And they preferred the 2019 research wines over the 2020. So their liking is driven by fruitiness, mainly stone fruit and tropical fruit flavors. And their detractors for liking were bitterness, with some of the lowest rating wines really having that high bitter value described. In Group 1B, they were driven by dry and complex flavors. So they had higher liking for one of the Vidal test yeast treatments, the IOC B-thiols, and they preferred that 2020 wine over the Vidal 1 and 2 wine. They had a liking score that was driven by low sweet taste and complex flavor. And their lowest rated wines in this group basically lacked the desirable characteristics that they were looking for in a wine. The third group, group number 2, had their liking driven by sweet taste and not sour. So they had a higher liking score for one of the Vidal commercial wines, Vidal 1, higher than our 2020 wines made by the B-thiols IOC yeast and the Vidal 3 wine. They had mid to low liking for most of the research wines. And this makes sense because detractors for this group were high acid and bitterness. And a lot of the research wines were fairly high in acid in both 2019 and 2020. So further to the consumer preference story, Dr. Bowen wanted to assess their willingness to pay. And so she presented them with a brief history of the Vidal grape grown in the region and what it's used for in wine production, and then carried out a test to look at their willingness to pay. And regardless of the liking group that was evaluated, the majority of the consumers were likely or somewhat likely to try a Vidal wine made in Ontario. So about 78% of the consumers said that they were likely or somewhat likely to try a Vidal wine. And that most consumers, 43% of them, would pay $13 to $16 for a bottle of Vidal table wine. And that's in this pie chart here on the right-hand side, where you can see the majority of consumers would be willing to pay $13 to $16 a bottle, which was higher than our target of $12 a bottle that we initially thought would be a target point for these wines. So with this initial research, I had some juice left over, and I had a 4th-year student, Dan Greaves, that was looking to do a project in the lab. And I really wanted to take a look at the impact of fermentation temperature and its impact on volatile thiol production in these Vidal wines. And this is coming from research, published research on Sauvignon Blanc, where it's known that higher fermentation temperatures are reported to increase volatile thiol concentration in Sauvignon Blanc. Also, just a few years prior to this research, I had been over having some wine with my friend and colleague, Carl Kaiser. Carl has since passed away, but he's co-founder of Inniskillen and was the winemaker there for many, many years. And Carl still made a lot of wine in his garage for his own consumption and the consumption of his family's. And one of the wine that he liked to make was Sauvignon Blanc. And we were sampling some Sauvignon Blanc, and he put a couple of bottles in front of me and said, "Here, try these wines." And one of the wines was just so full of aromatics that would be equated to the volatile phthals. And you know, I just commented to him that, wow, you know, this wine was just so aromatic and flavorful. You know, what did he do with this wine? And he chuckled and told me, well, on this particular batch that he was fermenting in the garage, the temperature got away from him, and the temperature spiked up to about 28 degrees Celsius. And he said, but you know what? In the end, it's the best Sauvignon Blanc that he ever made. And that really resonated with me, along with the data that had been published in the literature about the impact of temperature on the production of volatile phthals by the yeast. So we wanted to see whether temperature would have this impact in the Vidal wines. And we also wanted to further do some ferments without that stimula micronutrient and with the stimula micronutrient. We weren't able to do that in the original experimental design just because we had too many treatments already. But in this separate experiment, we carried that out. So we carried out fermentations at 14 degrees Celsius, so a cool white wine fermentation temperature, 20 degrees Celsius, just kind of a mid-range, non-stressful temperature for the yeast, and then a warm temperature of about 26 degrees Celsius. And we carried out these ferments using the stimula added to the juice and without the stimula added to the juice. And again, this was work carried out by my fourth-year thesis student, Dan Greaves. And when we carried out the fermentations, we saw that with or without the stimula, there was not a huge impact on viable cell concentration. Maybe allowing the cells to reach that peak cell concentration a little bit sooner with the stimula. And the stimula supplemented yeast at 14 degrees Celsius fermented a little bit faster, but you know, not equating to a huge difference, you know, that I think would be picked up in a commercial production. At 20 degrees Celsius, again, very similar results in cell growth and sugar consumption, maybe just slightly faster with the stimula, but really not much of a difference with and without the stimula. And at the 26 degrees of Celsius ferments, of course, the cells completed much faster. They reached their peak cell number faster, really not much of a difference with or without the stimula in terms of peak cell number or when they completed the ferment. But what happened when we started to look at the volatile phthals? So once again, we found 4 MMP and 3 MHA in the wines. And when we looked at the impact of temperature on the production of 4 MMP, we actually found whether the ferments were compared without the stimula or with the stimula, we did find that there was an increase in the 4 MMP. And without the stimula, there was actually about a 6-fold increase from the low temperature ferments to the higher temperature ferments. We saw a similar result in the 3 MHA, but just on a slightly lower scale. Interestingly, it was only at the 14 degrees C ferments that we actually saw a positive impact of the stimula increasing the 4 MMP concentration. At the higher temperature, we actually got a slightly lower concentration level of 4 MMP and also slightly lower concentration of the 3 MHA. So this definitely needs a little more research to further investigate the role of stimula. And part of this may also be the fact that we added DAP because the juices were a little bit low in nitrogen. And DAP may have impaired the ability of the yeast cells to take up that stimula by a phenomenon known in yeast as nitrogen catabolite repression. So we really need to repeat these experiments without that DAP addition prior to the stimula addition to really decipher where the stimula can make a difference. And once again, for both the 4-MMP and the 3-MHA, we saw similar values to what we were measuring in a commercial New Zealand Sauvignon Blanc. So in this fourth year thesis project, Dan really wanted to further analyze the volatile organic compounds that the yeast produced during the fermentation. But due to our COVID restrictions, we were just, there wasn't enough time in the lab to be able to carry out the VOC analysis. There also wasn't enough time to carry out the sensory analysis on the wines. So these are things we look forward to doing in the future. So to finish up and summarize, the Vidal table wines that we produced from grapes in the Niagara Peninsula did contain measurable levels of the 4-MMP and the 3-MHA, and the values were similar to what we were picking up in our control wines that we were testing. Crop load did not impact the volatile thiol concentration in the wines, but please keep in mind that we were only able to test that over one vintage, and it was a really cool vintage or cool growing season. So there may still be some impact that we just weren't able to pick up in our study. Conditions that can be manipulated in the winery, such as yeast strain choice, fermentation temperature, fruit ripeness, and nutrient additives, were found to alter the volatile thiol content in the Vidal table wines. A great maturity increase of 3 Brix did produce wines with higher volatile thiols, but a 1 degrees Brix increase did not impact the value in the wines. So in the future, target Brix levels may need to be closer to that 20 Brix value that we tested in 2020 versus the 18 Brix value we tested in 2019 and 2020 to further optimize the volatile thiols. That Sauvy yeast in this study produced more volatile thiols than the B thiols, the QA23 and the EC 1118 yeast. And you'll remember back from that slide earlier, this slide, had some descriptors that are commonly found from volatile thiols. The addition of the 26 degrees Celsius fermentations produced wines with the highest levels of the 3-MHA and 4-MMP over those measured at the lower 14 and 20 degrees C temperatures. So temperature does appear to increase the volatile thiols in Vidal as reported in Sauvignon Blanc. The addition of stimulus significantly increased the levels of 4-MMP in the wines fermented at 14 degrees Celsius, but actually decreased the value slightly at 26 degrees Celsius. So we need to further investigate that to see what's going on. And further enological treatment evaluation is required to assess the impact of treatments on the overall volatile organic compound profile in the wines. And for those temperature manipulated fermentations, we'd like to do some sensory descriptive analysis on those wines. Consumer preference testing provided valuable insights on guiding future research on these wines. Fruit forward and complex were drivers of liking, and high acid and bitterness were detractors of liking in Vidal table wine. So we can use this information in future, that if we have another high acid year, it'll be important for us to take the steps to further deacidify those wines before we finalize those wines. The Vidal research wines scored higher in liking scores than commercial Vidal wines in some consumer groups. And we saw that in the global overall liking scores, as well as when we funneled that down further into those 3 consumer groups. And the consumers indicated that there was a willingness to pay of about $13 to $16 per bottle of locally produced dry Vidal table wine. And I should also comment, I forgot to mention earlier, that the consumer panel was made up of over 100 people. The work carried out at Vineland, they were able to present these wines to over 100 people. So in closing, I'd just like to acknowledge the collaborators on this work, Dr. Amy Bowen from Vineland Research and Innovation Center, Dr. Jim Woolworth from the Department of Biological Sciences and Covey, Dr. Jennifer Kelly, postdoctoral fellow at Covey, and I just realized I'd left off Sufan Zhu, our Covey technologist that worked with us to develop the volatile thiol measurement technique. Dan Greaves, the fourth-year honors thesis student that did that latter work. And of course, Albert Zager, the grape grower that donated all the grapes and was provided industry input and was really a driver forward for this project to look at alternative wine styles for the Vidal grape in wine. Of course, we wouldn't have been able to do any of this work without the funding from Ontario Grape and Wine Research, Inc. And with that, I think I'm right on just maybe a minute or two over with the lecture. So I'd be happy to entertain any questions that might come up.
Well, thank you, Debbie, for that informative presentation. We'll now start the Q&A portion of the lecture. Our first question was, you mentioned sort of temperature variables and different preferences. What was the most interesting or unexpected discovery from this research?
Well, I think the most interesting discovery is that volatile thiols are present in Vidal table wine. We had a hunch that was the case just based on our own sensorial evaluation of some commercially produced Vidal wines, but that in and of itself was, really an important result from this work. We also, I was really quite intrigued that we were seeing that increase in the volatile thiols at the warmer temperature, and that further solidified in my mind, the discussions I had with Carl Kaiser about that glitch that happened in his garage Sauvignon Blanc. We seem to be repeating that in the Vidal. I think we can further investigate that temperature regime by maybe changing the profile during the fermentation, maybe allowing that temperature to spike up early in the fermentation to make those volatile thiols, but then, further altering that fermentation profile to a lower temperature to capture all of those other volatile organic compounds that we haven't had a chance to measure yet. I was also really intrigued that there were some striking differences in the various yeasts that we trialed. And we have some additional yeasts we'd like to trial that we think may further enhance the volatile thiol content. So lots of interesting results from this work, but it's led to many more questions for us down the road.
Great. We have another question that's come in. How important are volatile thiols in other wine grape varieties when it comes to profiles?
So yes, Sauvignon Blanc would be the most predominant, let's say, in terms of that volatile thiol profile. Often Sauvignon Blanc is described with attributes of on the fruity side of grapefruit and gooseberry and passion fruit. And that was, really what we were trying to further enhance in the Vidal. Sauvignon Blanc also, is known for contributions from methoxypyrazines. We didn't do those measurements in the Vidal, so I can't comment on that from this trial. But really, those volatile thiols show forward in aromatic whites like Sauvignon Blanc, and are really, one of the main descriptors when you hear people describe Sauvignon Blanc with those fruity characteristics. They're coming from those volatile thiols.
And where are you hoping to take this research from here?
So, you know, we found some really interesting results from this work, but I really want to look more into that temperature regime throughout the fermentation and see how that will impact the volatile thiols in the wine. I really need to further investigate the role of the stimula micronutrient additive that's supposed to contain these precursors to the volatile thiols that the yeast can then utilize. But I think we had a bit of a flaw in our experimental design to really see the impact of the stimula, in that we added diammonium phosphate to the starting juice. And in yeast, there's a process known as nitrogen catabolite repression, that when a really good nitrogen source like ammonia is added to the yeast, they don't allow the transport of some of these other nitrogen compounds, like these precursor compounds, into the yeast cell. And so we want to see, you know, really investigate the role of that stimula by not adding that DAP to the ferment, and seeing, you know, what the role of that stimula could be in further enhancing the volatile thiols. So, you know, as I said, there's also additional yeast strains we want to look at. We've got a locally isolated yeast that is known to enhance the aroma profile, the fruity profile, makes lots of acetate esters, you know, in the wines. And, you know, one of those volatile thiols is an acetate ester. So, yeah, we're just really intrigued by what this research has allowed us to start to investigate in Vidal table wine, and we think there are further permutations we can do to further enhance that aromatic profile and make a wine, you know, following the profile that the consumers indicated, you know, that they would be interested in. So I can't emphasize enough the value of that consumer insight research, you know, that we did to, you know, really help hone in what consumers are looking for. We know that now, and we can use that information to guide, you know, the future enological treatments that we carry out.
Great. Well, that looks to be all of our questions for today. Once again, I'd like to thank Debbie for joining us today, and to everyone who joined us virtually. Today's lecture video will be posted shortly to our website at brocu.ca/Covey. And you can also find more info about the lecture series as a whole on that website. That was the final lecture in the 2022 Covey Lecture Series. We look forward to seeing everyone again next year. Have a great day.